Okay, so now we basically are at what we want to talk about for the rest of the lecture. And so this is this RNA sequencing. So as I mentioned before, we always start with DNA, right? And so this is our base information. And then in a RNA-seq experiment, we have in vivo, meaning like in the samples themselves, we have the maturation of mRNA to mature mRNA, meaning that we go from DNA to a pre-mRNA, which is still including all the introns and all the exons. And then after splicing, we have a transcript, a mature mRNA. And then, so what we do is we extract all the mRNA from the cells. And then there's a step called fragmentation, which is basically using like a very, like an ultrasound or something, like it's like a sonication, which basically makes the mRNA shake or vibrate. And depending on the frequency, it will break the mRNA into pieces of like a desired length. So depending on the experiment, they usually try to break the mRNA into sequences of about 200 base pairs. So this is kind of the thing they aim for, and then you can use technologies like bioanalyzer to like see how long your sequences are, but this can be somewhat relevant if you align the sequences later. So it's good to know what the actual length of these fragments is, but so this is the sonification or fragmentation step. Then you do reverse transcription. So you basically want to build a cDNA library so it's more stable, and then you get your DNA fragments, and then you want to multiply those. So you create a you just go through a lot of steps of multiplication of these sequences, and then you run them through the sequence aligner. So this is the like an expensive machine, and it will output a FASTQ file. And we'll mention in a second how they exactly look like. But so this basically gives you a large text file of genetic sequences. And then the computational task, which is the in silico step, is to take one of these sequences and then know where they come from in the reference genome, right? And so this gets also complicated because the human reference genome is about 3 billion base pairs, right? But because, as you see here, we have the DNA sequence, right? But the DNA sequence is actually not the correct reference because the mRNA does get changed, right? So we cut out things. So there's all of a sudden like splicing interfaces that don't exist on the reference. Certain exons are missing, right? Like stuff like that. So the alignment algorithms have to be able to deal with this splicing variation. And then advanced techniques don't not only want to know what gene is at what level is expressed, but maybe they also want to know at the transcript level which transcript is expressed more than the other. So in this example, we can see that a lot of the reads were aligned to this first exon, and then fewer were aligned to this middle exon, and then more were again aligned to the last exon, suggesting that both transcripts were potentially expressed the same amount, right? This is kind of what this looks like, right? Are there any questions so far? No? Okay. Okay. So, yeah, so as I mentioned before, so this is quite a similar slide. There is, we have our RNA-seq reads, and then there is two ways we can deal with this. Either we use some kind of reference genome, which we have, right? Because we sequence more or less a lot of species nowadays, but like if it's human or mouse, there's full genomes. And then they also have annotations, right? So we know where the exons are and where the introns are, and we know what the possible transcripts are. So by using all this information, we don't have to, like come up with novel transcripts or something like that. We just know what we can find. So we can take the reference genome and we can build the, expected the splice junctions or sequences of possible transcripts, and then just align against those. Or there's like more complicated algorithms where you can do a de novo assembly of transcripts, which is much more complicated, and I don't think people use that much. But there's algorithms that can do this de novo. Like you would need a lot of RNA-seq data, like at a high depth. So meaning there's just like a high coverage over the whole transcriptome that you have, and then you could potentially figure out what the original like transcripts were, or what they looked like. So there's a nice paper if you want to look at that. You can. I have a slide in the back with all the references as well. Okay, so yeah, talking about the reference genomes and gene annotations, there is a couple, but I think the biggest one is this Ensemble website, and they have information about a lot of different species. So this is not just human or mouse. And so this is kind of how this looks like. And so this is for human. And so the important links are the download DNA sequence. So you can download a FASTA file, which is basically just a long text file with all the base pairs in a row. So it's just like a really large file. It's like, it's actually not that large. It's like 3 gigabytes if you unpack it. And it's 3 billion base pairs. So it's just ATC, GE, and like some ends maybe. And then the other important ones are on the right. So you see the cDNA sequences. So the cDNA sequences are um, the sequences of the transcripts. So you would get, if you look in the file, it's also a text file. Then you will get in the first line some information of what the sequence is from. So it could be a transcript, and it will say from what gene it is. And then it has the sequence of that transcript, and then it come like in the next line you get another transcript and so on. And then in the GTF file, that's just like a metadata file if you want, and it refers to the DNA sequence that you have on the left. And it will basically tell you where features can be found. So it will, for example, have, it will say chromosome 1, at location 1,200,000 something until 1,200,000, like something. And so then you know where exactly the sequence of that exon is, right? And then it will also tell you to what gene the exon belongs and stuff like this. So this GTF file is the genome annotation file, right? So here you will find all the information, where the transcripts are from, and it will also tell you which exon belongs to which transcript, and so on. Oh, there you go. Okay, so this is the GTF reference example. So this Havana is just a version name, but so this would mean that we have, this is the genome location, right? So like this is like 4 million or 40 million, 45 million to 45 million something. And it's an exon, right? And then you can see this is the gene ID, and then this is the transcript ID from Ensemble. And then this is the strand orientation, right? So this is kind of how this looks. Here's this transcript. So it basically this transcript length just embeds all the exons, right? So we go from the first exon beginning to the end of the last exon. So this is the sequence of the transcript. And then in there, you probably have all the introns as well, right? So here, because this exon, for example, goes to 5717, and then this one starts at 8295. So there's an intron in here with a couple of hundred base pairs, right? Okay. And then, yeah, what I wanted to show in this one is also that you can see that for different transcripts. So here we have two transcripts, right? This is the first and this is the second. They do belong to the same gene, right? But they also share some exons, right? But they have, so the red ones, they're duplicated, right? So these occur multiple times, but then the yellow ones are unique to these two transcripts. So this is one of the complications when you do RNA-seq alignment, is that the sequences are not unique for each transcript, right? And this is also true for genes. So oftentimes the sequences of a gene will be similar to another gene, because in evolution, how things develop is often a gene just gets duplicated, and then over millions of years they diverge in sequence, but they can maintain a lot of sequence similarity over time. So a lot of the reads will not uniquely map to one point on the genome. Okay, so this is the cDNA reference example that I was talking about. So this is two transcripts that we can see here. So this ENST is the ensemble transcript ID, and it also tells us what the gene ID it's from, and then also the reference genome. So this is for human 38, this is assembly, and so on. But then this is the sequence, and they always break them up into so that you don't have just a really long line. They break them up, let's say at 50 base pairs, and then they just keep going, right? So but you can concatenate them into one string, just to make it easier to look at. And you can see that did I just no, wait, what did I do? Yeah, so there's two different transcripts, and it's hard to see, but they probably share quite a bit of sequence similarity because they're from the same gene. And then, yeah, so just like for some stats, so in the human genome, there's about 200,000 annotated transcripts, and they are not all protein coding, right? So there's a lot of RNA products that will never turn into a protein. So that's something to keep in mind. So the amount of genes that actually code for protein coding things are much fewer than 40,000. So for protein coding genes, there's maybe 22,000 or something, 23,000 genes. But in the latest reference, there's 40,000 genes. And so these are the different lines. So we have an identifier, right, which always starts with an at symbol. And this is just the name of the read that comes from the sequencing machine. So this is the name that the machine gave this read. And this is the important thing. This is the sequence that it found in whatever was put in there. And then it has a identifier again, which I never understood. I do not understand why this line exists, because it takes up a lot of space and like it's completely superfluous, I think. But so there's a third line again with identifier, and then you have the quality score, and this quality score has the same length as the sequence up here, and they match, right? So they have the same length, and then the first entry here is the quality score of this read, or of this base pair, and then so on, right? And so this is called the Phred score. And it just basically tells you the confidence that this is really what was read in the sequencer. So if it gets unsure, like these are probably perfect scores, and then they start to degrade towards the end. So in general, with sequencing, the longer you make the reads, the higher your chance that you made a mistake. For example, maybe you slipped, like you missed one of the base pairs, and then everything afterwards is wrong. So that's why oftentimes the quality of the reads is good at the beginning, and then it can degrade over time. And then, so we have these four lines, and then we have another one. And so in a practical case, if you have fast Q data, we're talking about, let's say, 10 million reads up to, if it's single cell data, hundreds of millions of reads. Right. And then so these files are practically usually between 2 GB and 100 GB large. So pretty large files. So this is the Phred score as an example. There is different ways they can be encoded. So there is like Phred 33, and then there's like some other ones. But the idea is that we have a quality that usually goes to 40. I don't know why there's 42, but I think 40 is usually what the algorithms, like what the machine will output at best. So you can see here it's I, that was like what we had in our first example, right? All the reads were kind of perfect quality, meaning that our expected error is pretty low. And then as we go down this range, we get more and more sure that there's probably errors in our read. And so there's a nice tool called FastQC, which doesn't have a citation, otherwise I would have cited it, but there's a link that you can look at. And so here you can visualize the quality across all the reads given the position, right? So you can, in this example, see that at the beginning, the quality is really high. So we don't expect a lot of errors. And then the further we go into read, the higher the error gets. And then this is kind of a similar plot where we can see that the distribution of qualities scores is pretty high at the first base pair, and then it slowly degrades where it gets a little bit questionable, like if you want to use these reads in the end. But so this is generally something you can expect, that the quality, the longer the read gets, degrades slowly.